Morning, everyone. My name is Dr. Dick Walker. Welcome to my Saturday morning Redox for Medical Professionals webinar. This morning, I've got a great guest who's going to do the presentation, Dr. Lee Ostler from Tri-Cities, Washington. He's an expert on redox. In fact, he's written, as far as I'm concerned, the best book on redox called Redox Matters. It's at a level that's great for people with a little bit of science background, and it's great for people with a lot of science background because redox is such a new area, and there's some great information there that's quite pertinent to a SEER redox. So Lee's background is in dental science. He spent a lot of time putting this book together. I was a minor participant in reviewing some of the rough drafts and that sort of thing, but Lee's done a bang up job. If you haven't gotten that book, it's a great reference. It's not necessarily something that you sit down by the fireplace and read for entertainment, but if you want to know about redox in a really science practical sort of matter, it's a great resource. So Lee, welcome to Redox for Healthcare Professionals, and I'm going to turn it over to you because I know you've got a lot of material that you'd like to cover.
Thank you, Dr. Dick. It's my pleasure to be a part of the Redox for Health Professionals, or Redox for Docs, as it's sometimes called. And it's my pleasure to be here. I'm going to start my screen now, and we've got a rather formal presentation to present today. Redox for Docs, this is going to be about the science of redox signaling. We call it in the book, awakening the inner doctor with this redox science and cellular communication. The audience for today is, let's see. The audience for today is physicians, dentists, naturopaths, chiropractors, nutritionists, basically all health professionals, but also their patients, their clients, and those interested in cellular general health. This is the context of the book, Redox Matters. I put it together because I was being asked by a lot of physicians and dentists and other health professionals to show them the science. And that was kind of a hard thing to accomplish because there's a lack of science, at least the kind that they want to see in terms of random controlled trials and so forth. And if you want more information on the whys of that and kind of the background of that, there is material available. And anybody who's familiar with this company, the company is ASEA, or the beginning science of redox signaling molecules, can share that with you and explain why this is in the niche that it's in. And so that's where this came from, and that will be the nature of our discussion today. Is we want to get down to the very bottom of the redox triangle, if you will, and talk about the foundational issues of what the redox science really is. But first, let me just explain why I got into this. This had to do with a personal experience with my wife. In April of, this happened to be the 26th of April in 2018. This was a picture of her. She's had 30 years or 3 or 4 decades worth of ill health due to some pretty grave endocrine problems and metabolic problems. And we've been everywhere, all through the Northwest where we live, dozens and dozens of different physicians and different styles of medicine and healthcare. And it was in October of that year that our physician, Dr. Lynn, who many of you have seen, I'll show a picture of him here in just a moment, in a previous Redox for Health Professionals webcast, suggested to my wife and I that we get her on redox signaling molecules. And as I tell everybody, I remember looking at him and asking what that was, and he said, just go get on the internet and get some. And so not being satisfied with the answer, I did, and we did. And this was like 3 weeks later, the November 15th image. And about 3 weeks after that, 2.5, 3 weeks after that, this is what happened over 8 months' time to my wife. And when I when I looked at this and saw what was going on, I could not, you can't unlearn, you can't look a different way. My intense mind had to figure out why this was happening after all these years. And, if you want to hear a different version of this, this is just a couple screenshots from the Redox for Docs Health Professionals webcast dated 2 weeks ago, September 18th. It's available on vimeo.com/C United or the YouTube channel there. And Dr. Lin tells of this experience that we had through his eyes and what he was thinking. And that's a very interesting interview. You're going to want to go back and listen to that, as well as just the great things that he shared himself in that. So today we're going to be launching into a rather sciencey presentation of what redox signaling is. And redox signaling is, in the definition of it is in the name itself, the reduction oxidation. This is the story of how electrons transfer, or how we call it signal transduction or cellular communication. And so in understanding this, we have to begin at the very basics of this and talk about how electrons move and what an oxidant and a reductant agent is. And when you understand this concept and this principle, everything else kind of fits in. But if you miss this part, then you miss kind of what the foundation of redox biochemistry is. Everything in life is made of atoms, obviously. And these atoms are like a planetary system with a sun in the middle and planets, revolving around them. So in an atom, as you know, we have a proton or nucleus in the middle, and we have electrons that are rotating around it in their various orbits. And these orbits have energy, related to them. And they're very happily, living like this, except when an electron is lost or is stripped away from it, and then the molecule or the atom becomes very unstable. It has a higher amount of energy, and it now becomes oxidized. And so it's this loss of an electron that has been orbiting that creates an unstable situation where the molecule or atom becomes more reactive. And so the other side of the equation is again the molecule or atom that is missing the electron, that's looking for one. And so there's this constant exchange where a molecule that has an electron can donate or reduce a molecule that is missing the electron. And so this is the definition of what a reductant and an oxidant is. And as soon as a reductant gives up its electron or donates it to a oxidized molecule, thus making it stable, it itself, the former reductant, now becomes oxidized, and it seeks its own electron from another molecule. And this is a chain reaction that can go on and on and on and on. And it's all about energy gradients. Those oxidants that are more electronegative, as we call them, have greater force to attract or strip away or steal those electrons from reduced molecules. This is the basis, as we'll point out later, for the entire antioxidant industry that's been built up over the last few decades in providing plant-based reductants to provide these missing electrons to molecules or atoms in the body that are oxidized and can do damage. So if we kind of back up and we say, what's the big picture? What's this energy thing all about? Well, this energy is really about bioenergy. It's bioenergetics. All energy, as we know, comes from the sun, and it comes to our planet through the air in the form of photons or light energy that enters plants. And through a process called photosynthesis, the plant and the chlorophyll molecules inside the plant cells take that energy and combine it with carbon dioxide and produce the material, plant material, that is the structure of the plant, either in its roots or its leaves or its stems and so forth, in the form of stored energy. It's storing those in the form of these carbohydrates or fats or proteins. We then, mammals and humans, come along and we eat the plant, and this stored energy becomes available as fuel to be burned in our bodies in the form of either glycolysis or fatty acid metabolism, and they become these what we call Krebs intermediates and enter a Krebs cycle, combining with oxygen to perform a process of extracting energy out of that fuel or food source in a process called cellular respiration, which all occurs in the mitochondria. The carbon dioxide is the exhaust or the byproduct of that, and it enters back into the atmosphere. So we see this cycle where energy is circled or cycled or processed from the sun to the end points, which are mammals and all living organisms. If we dive into the mitochondria and look at that a little more closely, again, you can see these three fuel sources, energy sources of glucose or carbohydrates, free fatty acids, and amino acids and proteins. As you know, in Krebs cycle, the glucose is converted into pyruvate, which is then converted into acetyl-CoA. We also have free fatty acids that are made available from the breakdown of lipids in our body, and they enter this same process through a process called beta oxidation. So through both glycolysis and beta oxidation, we can take carbohydrates and lipids or fats and enter the Krebs cycle through this acetyl-CoA molecule. Amino acids also enter the Krebs cycle through several entry points into that cycle. Now, again, this is a busy slide, and this takes many of us back to our college days and for those that love biochemistry. But the point of this is, again, there at the top left, you've got the pyruvate and the acetyl-CoA coming into this. And if we took all of this reaction, a chain of reactions, and we laid it out on a flat line, as you see at the bottom, from citrate to oxaloacetate on the bottom, the point is that with each of these steps, each molecule in succession is becoming increasingly oxidized, which means we're taking things away. We're removing electrons, and in some cases, actual carbon molecules. And it's in the removal of those electrons that we're able to generate energy as it goes around. And then as it comes back to the end or the beginning, energy is put back into this through that process of glycolysis or beta oxidation. And it reforms the beginning molecule of citrate, which then starts this cycle all over again, and it goes over and over and over. And that's the basis of all energy that we have in our bodies. And that's how we harvest or take this fuel and make it available to us so that we can live and breathe and have our heart pump and so forth. So in this process of stripping these molecules away, we have these electron carriers called NADH and FADH2. Those are oxidized molecules to begin with, and they take the electrons away from the Krebs cycle intermediates and themselves become reduced. And now with those electrons available, they go through a cascade of reactions called the electron transport chain, which happens inside the mitochondria. And in each one of those steps, kind of think of it as a cascading waterfall. At each one of those, there is a removal of energy. The delta energy is removed, forming the ATP molecule, with oxygen at the very bottom, becoming the electron acceptor, because it is the most electronegative element, and it has this pull attracting these electrons down this cascading gradient, where the oxygen, which is oxidized, becomes reduced, and the byproduct of that is water. So that's an important slide, even though it's kind of a busy one, but this is the essence of life right here. And it's in this process, the other part of this process, because this describes glucose or carbohydrates and fatty acids, but the other part is the protein side of it. And we have several entry points in this cycle for amino acids to enter, and them also becoming a source of energy. So back to this oxygen. When oxygen receives these electrons, not 100% of the oxygen molecules are reduced. There's always about a 2 or 3% There's a range there of 1 to 5, but 2 or 3% of the oxygen molecules are not reduced, and they remain energetic, or we call them free radicals. So in addition to the metabolic or the Krebs cycle source for oxidized molecules, there's also oxidation that occurs as a consequence of toxins and what our environment is doing to us in the form of radiation or herbicides or pesticides and so forth. And those have an impact in the we call it complex 1. There's 5 complexes here that are going on in these reactions. And this is also producing oxidation, but it's also, in the consequence, it's reducing the ATP energy that is created. If we dive into the biochemistry of what these molecules are happening, the O2 or superoxide molecule that you'll see there in the red square is the electrophile, meaning that it is an electron-loving thing because it is an oxidant and it's looking for electrons. That becomes the prime player in this oxidation sequela. And if you can see on this reaction that the oxidized superoxide is in combination with peroxynitrate. When it combines with nitric oxide, it becomes super dihydrogen dioxide. Superoxide dismutase can catalyze it to hydrogen peroxide, and if we lack the glutathione there to the right, then it's shunted to become a hydroxyl reactant. So these are the oxidants, or the basic oxidants in this equation. The reductants are found in the superoxide dismutase and in the glutathione and the catalase that move these away from the superoxide and the hydrogen peroxide, forming water. But they also all have a signaling function. And so for decades, we've assumed that all of these free radicals or oxidants are bad. We've taken plant-based exogenous antioxidants to try and donate the reductant electrons to stabilize them. But we find now that they also have important signaling functions. And there's quite a class or a host of what we call cellular signaling messengers. We're just mostly focused here on those in the red dashed box here, the superoxide, hydrogen peroxide, hypochlorous, and nitric oxide. But we have to recognize these are just a small number of a broad class of signaling messengers that include transcription factors that we'll talk about in a moment. Also cytokines, chemokines, hormones, neurotransmitters, as well as charged metal ions. And so you'll hear, some people will comment that, well, you're missing the boat, and you're not talking about, metal ions, the calcium that have to do with channel blockers and so forth. Well, yes, that's true. There's a broad class of signaling messengers, but the really base of this are those that happen from this Krebs cycle and from the intermediates that are there. So with this basic, cellular biology about signaling messengers in place, it leads us to ask what I call the redox questions, and that is, how does a cell know it's under stress? What does it do about it? What happens in response? And we'll just assume that stress at this point means oxidation. That's the primary one, which can come from anything, including toxicity, radiation, hypoxia, or just the plain old Krebs cycle, superoxide. Not all of the electrons were captured by oxygen, and so we have these free radicals. So this is kind of the question that looms out there, is how does a cell know it's under stress? Well, this introduces a concept called the redox regulatory circuit, where there is something that happens, we'll call that a problem, and whatever that is, it needs to be detected or sensed in order to be able to generate a transduction or a form of communication that leads us to an effector, or what you would call the solution that turns into modulating the original problem that we had. So this cycle is really the philosophical concept and the construct between that relates to how does a cell or how does anything know there's a problem, and then what does it do about it. Many of us have seen this diagram many times, but this is essentially the same concept. when our cells are in homeostasis or they have balance, we're just fine. But when there's any kind of oxidative stress, a signal must be generated. There must be signal transduction or movement to the nucleus, where there's something that happens that generates a response that shows up with the necessary, processes and ingredients and so forth that solves the problem and puts us back into the oxidative balance. The means of whereby this happens are called transcription factors. These themselves are proteins that have their own genes that generate them and so forth, but these are utilized to generate the signals that control this genetic transcription and the production of mRNA, which is the process of translation that binds to a specific DNA sequence and begins that translation. And we're going to speak about two of them specifically today, called the NFKB and the NRF2. The nuclear factor kappa beta is a transcription factor that controls what we call the phase 1 cellular response. It happens when there is higher or sustained oxidative states. This is what the toxic response is all about. It's also involved in the initial or innate immune system response to things that go on in our environment, to injury or trauma or infection and so forth. It's also involved in chronic inflammation. And so when you have a kind of a low-grade, lingering, ongoing, sustained, stressful situation, you're going to provoke this phase 1 response, and it just goes on forever. And that's associated with much disease. On the other side, we have NRF2, which is the phase where it's representative of the phase 2 cellular response. It's mostly activated with low-level oxidation, and it is involved and responsible with activating the intra or inside cellular antioxidant response characteristic of glutathione, superoxide dismutase, catalase, and so forth. This is considered cytoprotective, cell protective. This is the defense system, the activating parts of the immune system. And in nature, it almost always accompanies the NFKB, with one exception, and that's actually not in nature, and that is relative to the discussion we're having with a supplement called ASEA Redox. We've determined in studies that you can see or access in other locations that when ASEA is administered as a supplement, it does not activate the phase 1 response, but it only activates the phase 2 response, which gives us a strategic advantage here, as I would call it a biohack, if you will, to be able to activate cellular protection. Now, what do these transcription factors do? And what they do is they translocate or move, they migrate to the nucleus, where they complex or bind onto the promoter regions of their specific mated genes, the DNA there, in locations that are called response elements. For the NRF2, we call those antioxidant response element. They've been renamed into electrophile response element. Those are synonymous and mean the same thing. And once that happens, then it can begin this mRNA process of translation. And so we see this kind of sequencing here, where we have the promoter region, which is activated, and it initiates this RNA translation in the ribosomes, where the mRNA is become active, and it translates, creating the amino acid chain of protein that then goes through post-translational modification as a protein that is folded because of the various charges on it. Again, those electrons that are in the respective molecules. Interestingly, these proteins can become disfigured because of oxidation. So when they lose an electron, it changes the charges in the protein molecule, and their shape will change, which means that their functional utility will change. That becomes pertinent as we start to look at, again, this process. This is a big picture process of this transcription and this translation process. You'll see on this, and I'll break this down a little more here in the future, or in the next slide, when there's a homeostasis situation or status there on the left, and this is showing NRF2, it is in conjunction with the keep one molecule, And so this keep one molecule becomes the molecule that binds with NRF2 to keep it stable. This is I would call it a dimer. So these two proteins are held together in normal homeostasis thing. The keep one molecule is rich with a thiol or sulfur containing thing called cysteine. And these cysteine residues, there's 27 of them on the keep one molecule, are very sensitive to oxidation. But when there's homeostasis, meaning no oxidation, these molecules last for about 20 minutes, then they degrade and they reform, and this is just continual turnover. In a state, however, of oxidation, when we have oxidants coming into the situation, producing more oxidation, they interact with these sensitive cysteine residues on the keep one molecule that changes their shape and thus releases the NRF2 molecule so that it can move or translocate to the nucleus, where it binds with that response element, the antioxidant response element, and it initiates that process of translation, doing mRNA production of these proteins that are the end result of this. This is the definition of gene expression. When this happens like this, when this NRF2 translocates and does this to the gene, this is called gene expression. We've activated NRF2 to have a pathway gene expression that produces an end result, or the effector that gives us the solution to our problem. If we jump over to the other side and look at the NFKB, the nuclear factor kappa beta, the same process is involved here. That molecule is held in check by a kinase inhibitor. And when there is oxidation present, it changes or degrades the kinase inhibitor, freeing up NFKB to translocate to its response element in the promoter region of the gene, whereby it begins the transcription and translation process, producing the end result, which are the inflammatory cytokines that are characteristic of this inflammatory pathway. So these are the mechanisms of action. Now, if we put this kind of back out and we look at it in a bigger picture, there's two models of what makes this upregulation of the transcription factor happen. And we're going to look at just this as an NFKB model right now. In this example, when there is a bacteria present, we see this in periodontal disease in the mouth. We see it in gut dysbiosis when you have bacteria, bad bacteria taking over in the gut. These and the endotoxins, or the lipopolysaccharides on the bacteria, they become the ligand that attract to a receptor called a toll-like receptor on the host cells that initiates this oxidation. And with this shift in the redox state, which is synonymous with the oxidative state, it affects this upregulation, as we just spoke to, of this NFKB producing the cytokines, which is then having the effect on cells in the form of matrix metalloproteinases, the osteoclast activation, adhesion molecules, chemotaxis, and a whole assortment of other things. This is the characteristic expression of the cytokine storm, for example, that we see with many diseases. The non-receptor mediated pathway doesn't involve a ligand or a lipopolysaccharide or a toll-like receptor. This is just the redox state inside the cell. When that oxidative stress happens via that the degradation of the kinase receptor or the keep one molecule, there's this shift in the redox state that produces oxidation. And that initiates the same NRF2 or the NFKB upregulation, producing the cytokines that produce the damage. So this explains why conditions such as sleep disorders, where there's hypoxia involved, or any other toxic situation that produces deficits internally, netting the net result being there's a hypoxia or any type of toxicity, as we mentioned earlier. All of these will shift that redox state, both producing free radicals themselves, which have a direct effect on cell damage because they can steal and manipulate the electrons and the charges on functional proteins that will change their shape or damage lipid membranes of cells and DNA and in the mitochondria. And so the point here is all of this is working together to activate the transcription factor activation that then has these net results of what's going on. So if we back up and we come back to the mitochondria, we say, what's going on in the mitochondria? Well, as you may have picked up, the phase 1 and the phase 2 is dependent on the concentration or the degree of oxidation there. This is a term that has been coined called the redox rheostat, and it has to do with the level or the concentration of oxidation dictates the outcome. You can think of this like an electrical circuit, this would be like a dimmer switch that will dim your lights. The outgoing energy current on this wire is a function not of the incoming current amount, but of where the sliding scale is on this coil of wire. This is what determines the amount of electricity or current that is able to, go forward. And in a biological sense, we have the same thing happening in the cell. This redox rheostat operates between the low oxidative state, some people have called that eustress, and the other end of the high oxidative state, or this would be the high distress. And this is a scale. It's not either on or off. It is a sliding scale, and the cell responds differently depending on where you are on the cell. If we're on the low side, we're going to have good high energy output. It'll be in homeostasis. Cells can proliferate and be healthy. If we're on the other end, we compromise our energy. We have low energy. We have oxidative damage that can lead to disease and cell death. And so this model is a great way of looking at how the cell works. And obviously, the ATP energy production is also a function of where we sit in this rheostat. Another way of describing this in the literature and in science is the concept of hormesis, or the hormetic curve. This is defined as an adaptive, biphasic, meaning it can go one of two ways, a response that results from exposure to a moderate, usually intermittent stress, such as with increasing amounts of a substance or a condition. In other words, this is a dose response where a single agent can have either a toxic or a beneficial response depending on the dose that is experienced. Again, the difference between eustress and distress. We see this example in pharmacology in the therapeutic window, microdosing, homeopathy. In physiology and nutrition, where we're looking at now, it's the difference between the toxic or the cell protective defensive response. And again, it's all about whether there's mild oxidation or excessive oxidation. Again, this rheostat concept. Pardon me. If we go back to this redox regulatory circuit and we layer this idea in, then we can see this come together where there's an oxidation event that NRF2, keep one, will sense and then translocate, which is the transduction or the movement, producing an antioxidant defense, which then modulates the initial oxidative state. Problem, detection, communication, solution. Layering that over our slide earlier, we see this concept of where the mitochondria is producing these reactive oxygen signals. Depending on their dosing or their concentration, we either get an NRF2 or an NFKB response. But there's several things here that we also need to layer in, and that is nature provides some solutions to this in the form of what we'll call bioenergy or redox signaling. And that has to do with anything that is nutritive, exercise, improved sleep, and so forth. These all have an effect on this equation because they enhance the mitochondrial production of reactive oxygen species, a subset of which are these redox signaling molecules that can come in and do this signaling based on their concentration. The unique thing here is that we can now introduce into this conversation, into this equation, a mild oxidative force or agent that particularly specifically targets the NRF2 transcription factor and the KEAP1 molecule, allowing for this phase 2 response selectively, to occur. We can call this or talk to this in the terms of a redox lifestyle that moves or balances this to the left. Again, expanding on this, it's important to remember or to learn that there's a lot of things in nature, as well as applied therapies, that will help us manipulate this formula or this sequence of events where the mitochondria become stimulated through anything that makes them more efficient or biogenic, meaning we're making more of them, the net result of which meaning that we're creating more reactive molecules that form these redox signaling molecules that then activate the transcription factors that then produce the end result, which helps to restore us to health. As I've listed here on the screen, you can see everything from ozone to exercise, magnetic, pulsed electromagnetic fields, hydrogen peroxide, both hot and cold lasers, low-level light therapies, vitamins, herbs, vitamin D, photobiotonic or photobiostimulation. All of these have an effect on health. If you ever wondered, well, it's like why do all of these things that you see, marketed, advertised, things that we can do, why do they make us healthy? Some may be better than others, but they all help move the needle in the direction of improved health. And the question is why? And this is the answer. Because they all have a positive effect on mitochondrial efficiency, either through more mitochondria or less oxidation and better utilization of that energy, which net results in the creation of more redox signaling molecules natively. This is already going on inside our bodies, trillions of times a second, just in the utilization of the Krebs cycle happening and normal living happening. And then, of course, with ASEA redox, we add something in addition to that, and we have something that's very synergistic going on. People say, well, where do you get your information? And I would just, as an aside, just send you, like I have, to the internet. There are thousands and thousands of articles out there, tens of thousands that talk about the science of these processes and how this is all working. And I would just encourage you, if you're intellectually honest and curious to know more about this, you can go into PubMed or in various search engines online. This is just some screenshots of several that support the mitochondrial support of all of these elements that we were just talking about. I want to illustrate with this whole process with pulling out of something I know very well about, and that's periodontal disease in the mouth. Out of this article, and this was written back in 2007, so this has been around just a little while. But even back then, we were seeing this concept of this redox rheostat, or this hormetic curve, hormesis concept, illustrated in the presentation of periodontal disease. Remember that periodontal disease is dealing with bacteria that have those lipopolysaccharide endotoxins that have that ligand specific effect on the cell. And when there's a lot of bacteria present, obviously the bacteria need to be removed. That's what having teeth cleanings and periodontal therapy is all about, as well as oral hygiene at home, as we're reducing the load, the bacterial load that is creating the oxidation in the tissues. But when we look at this from the tissue side, we see this redox rheostat. You can see the little teeter-totter there on the right of the screen, where we have reactive oxygen species representing oxidation, and we have antioxidants on the right. And depending on the status or the state of the cell, will determine where on the sliding scale the needle points. And so if we have too much oxidation, it'll tip down, and you can see there that on the bottom, we get the reactive oxygen species. Oxidation exceeds the defenses, so they're not adequate, or we're too overwhelmed, and we get the oxidant stress that produces the tissue damage. At the top, if I have more antioxidant protection in the cell is more in homeostasis, and that is an antioxidant or homeostatic state, and we can now do tissue repair. And then of course the variables in the middle would illustrate the anti-inflammatory state or the pro-inflammatory state with gradual protection or gradual destruction. So the point here is this overall redox balance within the cell is maintained by glutathione. That is the effector, that is the response, or the cytoprotective endogenous antioxidant protection that nature has designed to activate in the response to stress to keep the cells happy, healthy, and balanced. Oxidant stress as a result of excessive generation of oxidation within a cell or exposure to these exogenous sources of oxidation. They have the capacity, as I say here, to activate the redox sensitive signaling pathways, even in the absence of these specific bacteria, and in extreme instances can cause cell damage. So again, this concept has been around for a while. We're just now being able to put this all together. What this looks like, again, as we back up and look at the big picture, we all have bodies. The bodies are made of organs and tissues, which are made of cells, which involve cellular processes or subcellular metabolic pathways. Even below that, we have the genome, the DNA, genetics, chromosomes, and this genetic level is one that we've been highly interested in for many decades. We finally have the genome mapped now, and we talk about family history. We talk about 23andMe readings to be able to personalize medicine based on genetic propensities or defects that might be there. But the layer that's now below that is called epigenetics, and epigenetics is talking about the overlay or the influence upon the genetic layer. It's like what's going on at that subgenetic level, at the transcriptome level, that is influencing how our genes express good or bad. This introduces a concept called omics. We call it genomics and so forth. And this is about the different areas of the science. At the top, you see the metabolome. These metabolomes are the things that you can measure. The sugars, the nucleotides, the amino acids and proteins, the lipids. We have all these anytime you have blood drawn, very commonly you're measuring your blood sugar and your lipid profiles and so forth. That's the metabolome. Pardon me. Below that is the proteins or the proteome, and below that is what's happening at the RNA, which is the transcriptome. That's where the proteins come from, which is dictated by the DNA, which is the genome. You'll see on the left the phenotype top and the genotype at the bottom. We can't see the genotype. That's just your genetic coding. The phenotype is how the genes express. It's what you can visualize, see, you can test, you can measure, you can get lab values and so forth. And so we know a lot more about the phenotype than what's hidden down inside our genes. But the layer now that's coming in, as we talked about, is this epigenetic layer that's involved with the redox signaling. And this is called the redoxome. And when you hear people talk about redox signaling being a new category, this is a new category of health. This slide explains why that's the case, is because redox signaling is the undergirding activity or action or influence on everything else. And if you can get to the redoxome level and manipulate it and you can do that through diet, you can do that through exercise, you can do that through controlling your macronutrient, dinner plates of how much carbohydrates, proteins, and fats are on your plate, your frequency of eating, the amount of exercise you do. All of those things are epigenetic in their influence on mitochondria, which affects how much redox signaling is occurring, which determines how much cell protective immune system positive defense is going on in your body. So we see these side by side, again, from the body down to the epigenetics, and we can kind of pair these up and see a model develop here, where the redoxome is the foundation level. It's not the genes. It's what affects or influences or modifies the genes. Just because I might have a specific gene in my body that would predict or foretell, having cells go renegade or rogue on me and quickly start multiplying and dividing. That's not as important as, because you can't change that if you have those genes, but that's not as important as how they express, or if they express, or what we can do to keep them from expressing on the one hand, or on the healthy side, if we have processes that aren't happening that should be happening to defend and protect us, we can now manipulate them through this epigenetic layer or the redoxome layer of what happens at the mitochondrial level, and these transcription factors. We now have power and influence over there to affect a different outcome. Again, we see what's the phenotype, we see the genotype, and we can now talk about the epigenotype. And so this is the scientific model or concept here. I want to throw in one slide from the literature that illustrates this, and just point out the summary here that I've extracted from this article. The endogenous signaling molecules of the system, that means those that are inside, those are not exogenous, meaning we're not talking about plant-based antioxidants. We're talking about what the cell itself makes. They are mimicked by a wide range of chemicals and xenobiotics, that means foreign substances, that activate the system either by generating hydrogen peroxide via redox cycling, you see where it established that here, or simply by their ability to alkylate the thiols. Remember those thiols, those were the cysteine residues on the keep1 molecule, that when they disfigure through this alkylation process, it frees up the NRF2 protein to translocate to the nucleus. Going on here, such NRF2 activators, in particular the redox inert natural ones, are currently discussed for the safe adaptation to oxidative challenges and the prevention of related diseases. This is such a profound statement, because in one paragraph here, we have kind of the scientific talk of what we just laid out, step by step, as to how NRF2 activators become activated so that they can become set in motion the process that creates the effector, which modifies the original problem. The modification going on here of the NFKB, inflammatory, or the NRF2 antioxidant system has for long been, and still is, an attractive clinical perspective, in particular for the treatment of inflammatory diseases and the prevention of disease. So for the clinicians, looking in on this and involved in this, this provides a credible basis for the consideration of recommendations that are beyond, we'll say, the pharmacologic interventions, supporting the lifestyle modifications and even the use of a biohack in the form of a redox supplement, or anything that would activate these NRF2 activators. This is the basis, for those recommendations. The net effect here is, I've got this expression in the Redox Matters book, of activating the inner doctor. If you want to activate the inner doctor, what we just outlined in this presentation is the mechanism of action that credibilizes the entire redox signaling and mitochondrial activation pathways. And this empowers us to provide solutions that don't just act upon, such as a medical intervention, but allow the cell itself to do its own acting, restoring and recovering from within, because we're activating the very processes that the cell was designed to use to keep it safe. That concludes my presentation for today. If you want to learn more, all of this is outlined. Excuse me. All this is outlined in the book Redox Matters, which was just published here from the date of this recording a couple of months ago. It's become a great resource for many people, both health professional and otherwise. Again, it was written in response to physicians and health professionals asking the question of where's the science. I just remind everybody, in addressing that question of show me the science, we don't have the random controlled trials and the kind of science that we want to see duplicated 50 times over, because that's not the nature of what we're presenting here. This is a supplement ruled by the supplements rules on the FDA regulatory boards that govern supplements, and we can't make medical claims, and we don't talk about diseases. This is about cellular health and about awakening the inner doctor, but it's all about the redox biology. And so that's why the subtitle, Connecting the Dots Between Redox Biology and Health, is so important to understand, is in understanding the base epigenetic level redox biology. One can take that information and begin to extrapolate it and apply it to your life or the lives of your patients as an adjunctive measure to help improve basic health. More information can also be seen in terms of personal experiences at the realredoxresults.com website. The password there is Redox. The amazingmolecules.com website has 2 pages there with multiple videos and some PDF documents at the bottom of each page talking about the gene studies that were done, as well as some of the metabolomic studies that were done, which are very illustrative of this whole mitochondrial Krebs cycle effect there. You can also talk with the person that introduced you to Redox. So with that, I will unshare my screen and I'll turn it back over to Dr. Dick Walker and thank him for allowing me to have the floor and what I call the golden microphone for 45 minutes here. Thank you.
Well, Lee, thank you very much. That was a great presentation, and I think it's one that people will be sharing and using for a long time, because it's a very, very good explanation of why getting our redoxome tuned up and functioning as best it can, utilizing this supplement as an aid there is so important. And it's, you know, when we say this is a whole new science, a whole new area of science, and a whole new way of helping people regain their health, now people have a pretty firm understanding of how important it is and how it actually works. So again, thank you very much. I'm going to stop the recording here, but I really appreciate you doing this morning, and we'll keep up this series of scientific presentations for healthcare professionals and those who want to listen in. And again, this is not meant to cure, this supplement is not meant to cure, heal, or treat any specific illness. It's made to activate and energize your own inner doctor, which we all have at the cellular level. So thanks again.
Thank you.